Digestive system, AAQ Medical Science Unit 1. So, the digestive system, appearance and location of components of the digestive tract, the mouth and salivary gland, esophagus, stomach, duodenum, ileum, colon, rectum. Appearance and location of components of accessory organs, liver, gallbladder, and pancreas. Digestion, absorption, and assimilation of macronutrients, movement of food, bolus, or chyme through the digestive tract by peristalsis, the mouth, site of mechanical digestion by chewing, the role of saliva and salivary amylase in digesting starch maltose. Esophagus carries the food or bolus to the stomach. Stomach is a site of more mechanical digestion and chemical digestion and action of pepsin. The duodenum, action of bile, action of pancreatic enzymes, lipase, amylase, and trypsin. Ileum, site of completion of digestion and absorption of products of digestion. Structure and function of villi and microvilli. Assimilation of the products of digestion. Role of the liver in deamination of excess amino acids. Storage of glucose and glycogen. Role of amino acids, glucose and fatty acids in metabolism. Large intestine, the colon, its role in water absorption, importance of the gut microbiota. The rectum for storing feces, outline of disorders of the digestive system, gastric ulcers, irritable bowel syndrome, celiac disease, calculations relating to the digestive system, calculate rates of absorption and rates of enzyme action. So, the stomach is a large muscular sac that continues the mechanical and chemical digestion of food. Muscular walls churn the food to mechanically digest it. Contains hydrochloric acid to kill any bacteria ingested. This is also the optimum pH for protease enzymes which start protein digestion. The pancreas contains both exocrine glands which secrete digestive enzymes, endopeptidase, pancreatic amylase, and lipase into duodenum via the pancreatic duct. The endocrine glands that secrete insulin and glucagon into the blood to regulate the blood glucose levels. The gallbladder stores bile produced by liver and releases it via the bile duct. Then pancreatic duct into the duodenum. The bile has a pH of 8 to neutralise stomach acid. The duodenum is the first part of the small intestine. It's a site of neutralisation of stomach acid using the bile from the gallbladder. The ileum is the part of the small intestine, and the microvilli is the site of absorption of products of digestion, which then move into the bloodstream, glucose and amino acids or lymphatic system, lipids via chylomicrons are removed. Colon is the site of water absorption, and there's the role of gut bacteria in the large intestine. So the role is to ferment fibre, produces SCFAs for gut health, make vitamins, for example, K and B vitamins for metabolism, block pathogens, prevent harmful microbe growth, supports immunity, regulates immune responses, stabilises gut, maintains pH and lining integrity. So, where's that gone? These are the roles of the gut microbiota, the bacteria in the gut. Carbohydrate digestion starts in the mouth with amylase converting starch to maltose in the duodenum. Pancreatic amylase continues to hydrolyze the glycosidic bonds in starch to maltose. Maltose is then hydrolyzed to glucose by maltase enzymes embedded in the membrane of the epithelial cell lining the ileum. So you've got amylase produced in the salivary gland and pancreas, hydrolyzes starch to maltose. Maltase embedded in the epithelial cell membrane of the ileum hydrolyzes maltose to glucose. Starch digestion, so salivary amylase or starch to maltose. Pancreatic amylase is secreted into the small intestine, and then you've got brush border enzymes, maltase, which hydrolyzes maltose to glucose. Absorption of glucose, so sodium ions are actively transported out of the epithelial cells lining the ileum into the blood by the sodium-potassium pump. This creates a concentration of sodium, a higher concentration of sodium in the lumen of the ileum than in the epithelial cell. And then as sodium ions are carried into the epithelial cell via a co-transport carrier protein, they also carry glucose molecules with them, which creates a concentration gradient of glucose. There's a higher concentration of glucose in the epithelial cell than the blood. Glucose moves out of the cell into the blood by facilitated diffusion using a carrier protein. Villi structure. In the small intestine or ileum, there are finger-like projections which are covered in epithelial cells. It contains capillaries and a lacteal, and it's supported by connective tissue and smooth muscle. The function of the villi is to increase surface area for absorption, has capillaries to absorb glucose and amino acid, and it has lacteals in the middle to absorb fatty acids and glycerol. And then on the villi, the finger-like projections, each epithelial cells has even further small microvilli to increase surface area even further. So microvilli structure found on the epithelial cells covering each villus are tiny hair-like projections about 1 micrometer long. They together form a brush border. The microvilli function, they further increase the surface area for absorption, contain enzymes, for example maltase, sucrase for final digestion, transport proteins in membranes and move nutrients into the cell, specialised for active transport, diffusion and facilitated diffusion. Absorption of amino acids. Amino acids are absorbed in the same way, except instead of glucose, amino acids are moved instead. Protein digestion begins in the stomach with pepsin completed by a team of different enzymes in the lumen of the small intestine and epithelium of the small intestine, which break polypeptides into small peptides and then into amino acids. So endopeptidases, endo means inside, is an enzyme that breaks peptide bonds in the middle of the polypeptide into 2 smaller polypeptide fragments. Exopeptidases are a type of protease which hydrolyze the peptide bonds on the ends of the amino acids of the peptide molecules to chop off amino acids. And then dipeptidase splits dipeptides into single amino acids. So protein digestion, pepsin is produced in the stomach, has an optimum of pH 2, turns polypeptides into shorter polypeptides. Trypsin is secreted by the pancreas, its optimum is pH 8, functions in the duodenum, turns polypeptides into shorter polypeptides. Pancreas secretes exopeptidases into the duodenum, shorter polypeptides to amino acids. Dipeptidase is in the epithelial cell membrane of the ileum, and it hydrolyzes dipeptides into amino acids. Lipid digestion 1 enzyme lipase hydrolyzes the ester bonds in lipid molecules into glycerol and 3 fatty acids. Lipase comes from the pancreas and also the small intestine. So as a large lipid droplet, when you add bile salts, it emulsifies the lipids to make smaller lipid droplets, which increases the surface area, which means there's more surface area for lipase to act on to convert it into monoglycerides and fatty acids. Absorption of the products of lipid digestion. Monoglycerides and fatty acids diffuse out of micelles in the lumen into the epithelial cell. Monoglycerides and fatty acids recombine to triglycerides, which form globules, which become coated with protein to form chylomicrons. They leave via exocytosis and enter the lymphatic system. Assimilation, carbohydrate assimilation. At the liver or skeletal muscle, glucose is stored as glycogen. This is hydrolyzed to provide glucose for respiration. Excess glucose is converted to triglycerides and stored in adipose tissue. Protein assimilation. Amino acids are used throughout the body to synthesize proteins they need, for example, enzymes, antibodies, hormones, and transport proteins. Any excess amino acids are deaminated, releasing ammonia and then urea. Then it's removed by the kidneys. Lipid assimilation is transported as chylomicrons into lacteals. The body can also use the triglycerides for respiration by gluconeogenesis. Excess triglycerides are stored in adipose tissue. Roles in metabolism. Amino acids are used for protein synthesis or deaminated for energy. Glucose is a primary energy source via cellular respiration. Fatty acids are stored as triglycerides or used in aerobic respiration for energy. So summary, amylase is produced in the salivary gland, acts in the mouth, a pH of 7 optimum. Amylase is also secreted by the pancreas into the duodenum, pH optimum of 8. Lipase secreted by the pancreas into the duodenum, pH optimum of 8. Pepsin is secreted in the stomach, acts in the stomach, pH optimum of 2. Trypsin is released from the pancreas, acts in the duodenum, pH optimum of 8. Now looking at digestive system disorders, we've got gastric ulcers. These are sores in the stomach lining caused by H. pylori bacteria or long-time use of NSAIDs, like ibuprofen. Symptoms are stomach pain, indigestion, treatment, antibiotics and acid-reducing medication. Then you've got irritable bowel syndrome or IBS, a long-term gut condition with no visible damage. Symptoms are abdominal pain, bloating, diarrhea or constipation. Management is diet, so a low FODMAP diet, stress management and medication. 3 is celiac disease, autoimmune reaction to gluten that damages the small intestine. Symptoms are diarrhea, fatigue, weight loss, nutrient deficiencies. Treatment is strict gluten-free diet. So rate of absorption. Formula is the rate equals the amount absorbed over time. And usually the units are grams per minute. So if 60 grams were absorbed in 30 minutes, then it would just be 60 over 30 to get 2 grams per minute. Rate of enzyme action. Formula rate is product formed over time, usually in grams per second or milliliters per minute. So if 10 milliliters of a product was formed in 5 minutes, you'd just be 10 over 5 to get 2 milliliters per minute. Measuring enzyme catalyzed reactions, measure the product formed or the substrate used at intervals, and the rate is the gradient of the graph, so product over time. The initial rate is highest at the start because there's more substrates, so there's more collisions. The Vmax is where all enzyme active sites are occupied. The rate slows as substrate decreases, and a limiting factor is a variable that prevents Vmax increasing. It will raise the rate. That's the end of that section.